Have you ever wondered how viruses like those displayed in the TV commercials of disinfecting spray multiply? A lot of us may only know that they are just microscopic balls or capsule-like structures that can be found on different sites and surfaces. But it takes more than that on how they evolve and replicate. A virus, specifically referred in the field of virology as virion, is a single infectious particle that consists of either DNA or RNA genome enclosed in a protein-made covering termed as capsid. The process of replication is a series of steps and may vary according to type. To give an overview, it consists of 5 general steps. First is adsorption. It is basically when the virus attaches to a host receptor. Next, penetration is when the capsid is disassembling and the genome is being released. This is otherwise known as uncoating. Next is replication and translation. With the help of different enzymes, the genomes are coded and the proteins are produced. Assembly or maturation occurs afterwards. This is the phase where viral particles associate with others to form functional or matured virions. Then, it will be ready for release. The matured viral particles now exit the host cell and infect other cells. Let's dig deeper and observe the replication process in each type of virus between RNA and DNA viruses. Viral DNA replication starts with the attachment of the virion to a cellular receptor on the cell surface. Then it will proceed to the penetration to the host cell. Once it is inside the cell, it will travel to the nucleus through the microtubules. The shedding of the capsid happens inside the nucleus to release the genetic material. The DNA utilizes DNA-dependent RNA polymerase to initiate genome replication and transcription. Upon transcription to messenger RNA, the mRNA transfers to the cytoplasm. It is then translated into proteins with the help of the ribosomes. The translated proteins will be brought back to the nucleus where maturation to virion happens. Now, the release of the viral particles is dependent on the presence of the envelope. Enveloped viruses exit the cell through exocytosis or budding. The naked viruses makes the cell undergo stellolysis to exit from it. We will see how positive sense-strand RNA enveloped viruses, such as dengue virus, replicate inside the host cell. The viral particle first attached to the receptor at the surface of the host cell membrane. Due to selective permeability of the host cell membrane, viruses utilize the membrane proteins and enter the cell through endocytosis. Upon entry, it is internalized in an endosome. Since the genetic material of the virion is required to enter the cytoplasm, the capsid will be released in the cytoplasm after stimulation by lowered pH by proton pumps and conformation changes of envelope proteins, turning them to spikes and leading to the opening of the endosome. Then, the capsid will shed off in the cytoplasm and the genome will be released. The positive sense-strand RNA can act directly as a messenger RNA and read by ribosomes. The RNA can also be used already in protein synthesis or translation. In hybridization state, this positive sense-strand RNA can produce more RNAs, which can serve as templates for protein production and in maturation of new viral particles. Once it reaches a sufficient amount of proteins and RNA, it assembles itself and gets packaged in preparation to be a mature virion. The maturation also depends on how the virion exits the host cell. The packaged RNA and proteins undergo budding. The viral proteins are incorporated into the host cell's membrane and travel through the Golgi apparatus and endoplasmic reticulum while undergoing maturation. Once the mature virion has exited the cell, it will now infect the other cells. On the other hand, the negative sense-strand RNA is not detected by ribosomes. It should be transformed first into a positive sense-strand RNA. Termed as reverse transcription, the RNA strand uses its RNA-dependent RNA polymerase and transcriptase during the process to create more positive sense-strand RNAs. Once it turns to a positive sense-strand RNA, it will go through the same process. This time, there will be production of more negative sense-strand from the positive sense-strand RNA that will serve as genetic material. Then, it will proceed to the maturation process, which also happens in the endoplasmic reticulum and Golgi apparatus, and then exits from the host cell.